All right, so it's my pleasure to introduce today Eric Westhof, who is a well-known structural biologist with a very rich career in the RNA structure. So Eric got his PhD in Germany, if I remember well, and had many titles. Notably now he's also a member of the French and German Academy of Science. He was working in Strasbourg when he made many major discoveries, in notably the one I'm most interested in here, like the classification of base pair and all and found some structural model for RNAs. Eric now is still in Strasbourg, but also works at the Wenzhou Institute in China. So he's at both sides of the world simultaneously. And he made us the honors of visiting McGill this week and give us a visit and introducing one project that has been actually, you have been working for a very long time, that's called the RNA-Puzzle, which is a cast-like competition, but better, for assessing the progress that we're doing in RNA 3D structure predictions. You may think that we're done with structure with proteins. I mean, you all know it's not completely true, but in RNA, we're even much at the infancy of the problem. So it's very important to set a solid foundation and a scientifically rigorous framework for assessing how we're going forward and what we're missing and what should be done. And part of all this story will be told to you by Eric right now. And I thank you again very much for being here today. And I'm looking forward to hearing your talk.
I would like to talk about RNA-Puzzles and a little bit about RNA modeling, a little bit the history of it, how long it took, how long it took to work and build up. RNA modeling is not new. It's I think that is also important to us. So just to remember very basic stuff. The RNA is not protein. All right, you have only 4 bases, A, G, C, U. They are really remarkable, chemically speaking. You have 20 on the other side. Every nucleotide is, the bases are neutral, but every nucleotide is negatively charged. So it's a major difference between protein. Every single, you have a 400 nucleotide ribozyme that is 400 negative charges. This is a nightmare for computing that, the electrostatics. So the proteins, the alpha, beta sheets, etc., is H bonds between the backbone atoms. But what is fantastic in RNA is that the H bonds are between the bases, between the side chains. So that means the side chains tell you something about what is going on in the structure. So you can tell not only what's on pairs in double-stranded helices, but you have many others. I will come to this. Now, if you want to see only what we call secondary structure, that is the helices, you only need monovalent ions, sodium, potassium. You can see them, you can observe them. If you want to go to more intricate 3D structures, you will need magnesium ions. And magnesium ion is the very RNA without magnesium ion is not RNA anymore. No RNA works properly in cells without. You don't have to have very tight binding. We can stick, but it must be there. And the folding also is driven not only because you need these divalent ions, but also it's not the hydrophobic effect, but stacking of bases is might be related to hydrophobic effect, but it's a different type of physical mechanism. But anyway, stacking of bases is really a driving force. And coaxial stacking of helices, naturally, once you have an helix, you coaxially stack them or you parallel pack them, then you have non-Austrian base pairs. And non-Austrian base pairs are critical for all the specificity of RNA fold. And it's involved in selecting proteins, selecting a small molecule, selecting even the ribozyme, everywhere at the core of the function you see in the non-Austrian pairs. This, I think this testament. Now, again, these nucleotides, you have this beautiful, very nice backbone. Okay, you have 7 torsion angles. Naturally, that is much more than proteins, but we'll come back to this. But what is you have to remember is the ribose undergo pseudo rotation. That is, it can change states. And it can only be C2 prime, C3 prime, it can be in many states. And you need this mobility to unfold and fold back, you know. So without this, it would be much too rigid. And then that's an important point to remember. The phosphates are negatively charged. I mentioned that also. That's also very important and a big nightmare for all those who try to inject RNA into cells. But it is a critical point. And because these anionic phosphate oxygens, they are basically, they have partial charges. They attract water, they attract ions, so they are very special. And the bases, they are really special. They are the 4 purine and pyrimidine bases. These bases have a strong preference for neutral and tautomeric forms. It's rather unique in this respect. All right, just go back to the basics again. So you have the CC prime endo, where the CC prime is going up towards the 5 prime, as you see at the top here. This is the most common conformation in helices. And then what is fantastic in this nucleotide is that they reinforce each other. So if you have the CC prime endo, you will reinforce the anti-conformation of the base, like here. And then you have this O5 prime will be gauche plus, as we say, that is in front of the CH here. So all this conformation reinforce each other and make this CC prime endo conformer really the preferred one and the present helices. But you need the other one if you want to make a complex structure, because when you want to get out of an helix or when you have in kinks or in loops, you need to do C2 prime endo, and then the conditions are different. All right. Now, just to see, you have 7 torsional angles, but they are highly preferred regions. You don't have to scan all of the torsional angles, all the values. You see, you have gauche minus, the helices is gauche minus, but naturally, if you are in the helices, you will find mainly here, but if you are in single strand, you see all the stuff. And then this, the O5 prime and C3 prime are basically in primes, 180 degrees. This one is generally gauche plus, but you do have also other ones in loops. And you have the C3 prime endo, and massively C2 prime endo also in some loops, etc. So they are highly preferred. But still, despite these preferences, slight changes here and there allow you for a great diversity in folding. All right, a quick time on RNA modeling. So I told you it's not new. So the first one was the anticodon loop, 1967. Fuller and Bishop, they were fascinated by the loops. It was known already there, just barely, and that it had to turn. It has to present the anticodon for bearing to the codon, and they presented that in a paper in Nature. And in 1969, Michael Levitt proposed the structure of tRNA. It's a very interesting structure. It was not quite right, but it was still, it made a big impact. Then you have a big gap, until some times later, when in 1985. And during all these days, I will show you, there was nothing known about RNA structure except for tRNA, basically. And so in 1990, we have the problem, the group 1 intron, core of group 1 intron. And then we continued several of those different type of structure required. Then I started RNA-Puzzles in 2011. And since then, we have been doing less modeling, but trying to develop tools to assess and develop new programs. But the others, those are really good in bioinformatics and computer science. So that is the model of I don't want to go there in detail, but it's really interesting where he made the mistakes and how it is to relate to the tRNA structure. Now, that work on the group 1 intron, that's what made possible, and I would insist a lot, because we worked with sequence alignment, very precise sequence alignments. We had a lot of sequence alignment group 1 introns, and so you knew what was, and you aligned very precisely. That is, hours and hours are aligned. And then when you build in 3D space, comforting with those sequences, you can really find different type of contacts and to maintain this. Because when you have decided that these 2 helices are on top of each other, they are there, they are not down there, etc. And this leads naturally to other consequences for the rest of the room. So that was really already, that was a long time ago. You see, the RMSD is 15 angstrom for 4,000 angstrom. That is the last one from cryo-EM, 2001 from Judas and Wachu, etc. in Stanford, right? So this is a number you could keep to see what people are reached now, right? And this is another group on intron. This is Azoarchus. That was already better, it's smaller, 7 angstrom, but that was also much later. That was 20 years afterwards, or 10 years afterwards. Now, what is really also important to understand is that the RNA structures, the amount of RNA structures available for a long time, only tRNA. You see, the ribosome amyloid is 1995. Right? So it's basically, it's not very long. And then suddenly the structure moved on, etc. Tetramer, Jennifer, etc. made a big difference. And so it started increases. And then at some point, naturally, you ended up with the ribosome in 2000. That makes a massive impact because the ribosome is so rich in structures. All right, no, a modeling tool. So this is also interesting. The first one were in 1978. We used Frodo, Alvin Jones. That was the program mainly used for fitting protein in density, but we had implemented all the tools necessary to handle RNA. Nuclear was also developed that to compare to protein and pro-LSQ from Wayne Hendrickson. So that was the only program for refining RNA structures. Then Midas, MCC for Professor Major, Chimera was better, the S2S also from Sure, and then Farfar from Redutas, MC-Fold, iFold mode RNA composers, etc. from the Polish groups, and SimRNA, VFold, etc. from CJHN, etc. So all the programs have been adding up and we start, right? So now you have a little bit the history, the framework, etc. RNA-Puzzles. So we started doing this because there were more and more models and more programs and people didn't know where to go. And so it was at the RNA meeting. So we decided to do that collectively. And it's a collective blind experiments for the evaluation of automatic RNA experimental structures. So for this, I need RNA structures, you need structures on one side, and you need modelers also on the other side, right? And they say it's blind there because also a difference from CASP, when we publish a sequence to be modeled, we don't say where it comes from. So the modelers do not know who did the structure. You don't want them to ask whatever, this is also RNA structures. When we started to where if you publish that, you have a new RNA structure, you have in Nature, right? Or in Science or in Cell. So nowadays you have a new RNA structure. It's difficult to get there. Huh? So it was big competition. So we have to be very careful with confidentiality. That's the main point. So we started with Jose Cruz, who was PhD from Lisbon with me then, and then I continued now with Chicha, who was a postdoc in Strasbourg, and then now he's in Guangzhou, and so we can work, and it's not too far from where I am from time to time. All right, so that is the progress and the different meeting we had, etc. And after the success of AlphaFold, the CAS people became interested in RNA, and so they asked me to participate. So we participated in 2022 with CAS15 in different ways. So, and we participated also last year in CAS16. And we have the results. So I will present a mixture of the Fan et al. Paper and the recent one from the CAS16. So the difference with CAS is that we work on we accept puzzles on a rolling basis. That is constantly, because if somebody has a structure, you don't want to wait that he has the structure only in the period where, in the 3 months where you accept structures, you want all the time. So we accept it, and then naturally deadlines, everything's are fixed with him, with her, with the group, so that they can tell that when they will publish it, right? So that means that there is a little disadvantage. There is a time span between the time we put all the papers together and the result, but that can be reduced. And we publish the papers together between the structurers and the modelers, etc. So that everybody is there because they contributed all to this approach. And so with CASP, I don't know whether we will continue or not, but it's not important. Our approach is rather different. All right, naturally, because we are dealing with structures, RNA structures from X-ray or more and more now cryo-EM, you have to have a cryo-EM structure, or you have to have crystals, whatever. That means you have to have the biochemistry, you have to be able to have a stable fold so that they can be observed experimentally, even cryo-EM. If it is too dynamics, you will not see anything. And even if you take a random, you have a RFAM sequence, you take one sequence there, maybe that one will crystallize, but maybe the next one will crystallize much better, or probably the one before will not crystallize at all. So some are propitious for crystallizing, but not all of them. But still, all of them have the same type of structure, but they are stabilized in different states, in different conditions, right? So not every sequence will give a fault. So we have to try many more. That's why also sequence alignments are so powerful, so important. And naturally, we don't have the picosecond dynamics. We can see the molecular dynamics as is seen in NMR and all these kind of things, but that's we can discuss that more. All right, so here, for example, here you have some of those recent ones. The puzzles, you see different sizes, smaller ones, bigger ones, etc. And I put here the RMSD because RMSD has a lot of disadvantages, but it's a simple measure and you can understand very easily what it means. So sometimes you see naturally, you see, for example, 2.6 here, right, very good. But sometimes even in the ribo switches, it goes up to 3.4. Here in the ribozyme, naturally, they are a little bit larger, but you are 9 angstrom, 21 angstrom, 29 or 28, something like that. So you have a lot of variation. Here, 5, 11, T-box. Even the T-box, the RMSD is much better because of the tRNA, because tRNA, everybody can model a tRNA structure. But now tRNA with a protein or tRNA with another RNA, that's something else. But the RMSD will be dominated by the tRNA. So we have a great variety. And now if you look, you see, well, you see the fold, you see the green and the blue. The blue is the best, and the green is the experimental structure. You see, okay, globally, sometimes the fault is not too bad. You see, even when you are like here, at close to 5 angstrom, you see that you recognize the fault in both of them. But now look here, you see already that all the this are supposed to be the basis. They are all clearly in. So we want to see what is inside. So, all right, you get the global fault, you get a rough positioning of helices, but what is inside? All right, and I will say. So the appears history, but that's the question. And I think it's very important to really realize that the global fault is not sufficient. And it can be even deceptive to capture the key 3D features. These 3D features, they are in charge, responsible for the function of that RNA and the architecture. And if you want to explain the biological function, you have to understand all those internal bases. What do they do with each other? And why are they maintained? Why are they conserved, and etc. So, and this, I come back to this sequence alignment. This is just a sequence alignment from tRNA. And you can really see that the yellow, they pair together, and the colors, they pair together, the purple pair together, the cyan pair together. Here, this is the antigon loop. It's not too much. I can take any one, but tRNA, where is it? Where is the, there. Yeah, this is the antigon loop. But you see very clearly also that between the green helix and the cyan, here is this sharp. There is one nucleotide, no variation. Here between the yellow and the green helix, there are 2 nucleotides, no variation. The only variations occur in this region, but there you see the 2 GGs are always present, all tRNA have this, etc. So you have regions here, it's very variable, but here again, it's very rigid. Between the purple and yellow, it's sharp. There is never a new additional nucleotide. This is massive information for the 3D. And here, this is the T loop. You have even conservation. It's always the purple. It always ends up with a G and start again with a C. It's the G and the C in all tRNA, in all 3 kingdoms of life. Wherever you are, except in mitochondria, maybe. But in normal cells, cytoplasm, it's always like this. All right? Even when you have a T-like loop in a ribosome or wherever. So the GC pair is conserved. So when you have this information, that is extremely vicious, it means something. And we know exactly now why, which kind of base pair, whatever, etc. But this, if you can, the idea is that if you predict something, you have to be able to predict a sequence alignment. You have to be, okay, I can explain to you why these GG are conserved, why this A is always at this position, etc. And why this GC is that I don't, do you need to explain this? Because that's the only explanation you are looking for. Do you want to explain what is conserved, what is semi-conserved, what is not conserved? And naturally, the covariation between the helices, that is easier. Right, I will show you an example. Here, this is a Glenn riboswitch. And so it's an interesting structure. When you have a G bulge here, you see with an hosting base pairs, I will come back to this. You have also this single-stranded making a tertiary type of interactions. And you see a blend is captured between all sorts of non-hosting base pairs. This one is a CC and Watson-Crick pair, that is a normal one. But it is really very close to HTCR. So this is the blend. Now, what is very amazing in this structure, all these G signs is very rare. That's very rare. And this would be very tough to predict. That's really an exception. And now, if you compare. Now you can compare. So see, again, I put compared. So the yellow is X-ray and the blue is the structure. I took one, the RMSD 7.6 angstrom. So you see globally, okay, they got it right, a little the stacking, etc. But suppose if you want to look at a base pair G23C20, that's this in yellow. They're really touching each other. They are here, you see. But in the middle, one is over there and the other one is over there. They don't see each other. So it's totally missed. Right? They're really far apart, despite the fact that the fault is globally rare. Now I can look at another one, A6C21. That is this one. That is very key because it is in between these two helices, and this one is you go all this way around, you come this, and you go out. So this one is very key to maintain this coaxial stack. You see the red one is here, the touch is all, but you see that in model they are really far apart again. So again, they didn't catch this really critical interaction for the fault. Right? Despite the fact that globally they have to. So you don't expect them to have it exactly the same year, but nobody cares really about what is going on there in this big loops. And again, we know that from sequence alignment, you can align that extremely precisely. You see aqua and GAA, the G loop like is always there. So if you miss this one, what can you do? You see also you see exactly where the variations are, where they are not, and what is really always superimposable. So even if you don't know the structure, you can still deduce that you could get this. You cannot explain what is interacting with what. That would be difficult, but you can align so that you have no gaps here, you have no gaps so that the loop E is overemphasized, etc. This you can do by alignment, right? So I have to go back to the non-basotic base pairs and the work with nucleus. And so again, you have many possibilities. If you look at the 8 bases like edges, you have 3 edges: the Watson-Crick edge, the Hoogstien edge, and the Sugar edge. And then you can do the combinatorics. And naturally, you have the Watson-Crick insist that will be anti-parallel, that the 2D structures. These are helices, right? Fine. But then you have 11 others. All those, they occur in 3D structures. It's basically impossible to have a 3D structure, a complex 3D structure without any of them, right? So if the simplest four-way junction, yes, maybe, all right? But otherwise, even there, you see the choice for the four-way junction is made by the interaction between these two loops. The same here, the choice between these three-way junction is made by interaction between these two loops. And these interactions are not Watson-Crick. But what made the specificity of the fault here is the interaction between and non-Watson interaction between these two loops. Right? So all those occur. What is important also, you know that helices are anti-parallel, but here you see your parallel, anti-parallel, anti-parallel. Some of them are strands are anti-parallel, some of them are parallel. And you need that because it's a single strand folding in space. And if you have a single strand and you fold it in space, sometimes it will be parallel. No way. It's impossible to have only anti-parallel region. And that's why you need this non-Watson base pair, precisely. And at the same time, you follow the strand, the direction of the strand, but you also have stacking between the strands, even if you change the stacking. And you open regions. For example, if you make sugar H Hoogstien in France, like here, a very famous one. This is called also the shared base pair. This nomenclature is here. You don't have to have poetic name. You just look. The name gives you the base pair. You only have to remember how a nucleotide looks like, right? If I say oxytocin of one, and the sugar of the other one. It's like this, all right? And then you have to be cis or trans. That is, the sugar are there on the same side or on the opposite side of the line of interaction. So you give the name, you have to structure it. Only you have to remember one nucleotide. It's not too much. But that means that in this case, for example, if you give the oxytocin edge, then actually the Watson crick edge is free. Then the drug can come. A protein side chain can interact with the Watson crick edge, and protein side chain, but you prefer Watson crick edge than any other way. All right, but you are still inside the helix and you are still packing together. But you allow for different interaction. They are different than the usual interactions that you can see in DNA protein interactions, where they interact only with the oxytocin side. And that is why some time ago with François Major, etc., and Parisien, we developed this matrix that is what we call the interaction network fidelity. So when we look at a structure, we want to know not only where are the Watson crick base pairs, that is the 2D structures, the blue ones, you have to count them, you shouldn't miss some. We want to know where are the non-Watson crick base pairs, did you identify them correctly and where they are? And then we want to know who is stacking to what, on which, all right? Because this is very important. Because if you have a three-way junction, if this helix stacks with this one, or if the helix stacks with that one, the 3D will be very different. But that means here the CC is stacking under the UA, and here this one, the IU, what's so different. So you want to know that. And this is what we have access. And the idea is that if you have a good structure prediction, you have a correct interaction network, basically. More you have errors in the interaction network, the worse you go away from the real structure. All right. So, and then you can, the way it is computed is what's his name again? Comment il s'appelle encore ce truc?
Matthews coefficient.
Oui, Matthews coefficient, voilà. Elle est trop positif, les phases positives, etc. Vous savez ça mieux que moi. Donc, voilà. Donc, on va calculer, all this will be computed and systematically. Then we develop also something different, that is the deformation profile. Because we want to know, so what is, when you superimpose two molecules, we want to know where are the most discrepancies between the two molecules. Right, so in this case, you superimpose 5 nucleotides by 5 nucleotides, and you see how far away are all the other ones. And so you make that in a 2x2 matrix. I will show you an example, it's easier. Like here, for example, you have the sequence horizontally and vertically, and you see that all this region, for example, is badly. So this region here, and you see the B factor here, so this is dynamic. So you understand. So these dynamic regions, they are never in real agreement with the rest of the core of the molecule. Right? But these regions, they are okay, happy with everybody, etc. So you see which region are really in a good agreement with the rest of the molecule. All right. So this is for final analysis. So now I can show you boring stuff like this, Spearman's coefficient and all this, and then IDT, info, class score. So we look at some of them, all right? But they can do a lot of statistics there and try to see, for example, which ones are interesting and which ones are not interesting. So in all the way it is computing, you should be close to 1, right? That is, if you have it always should be small. TM score, it should be also close to 1. So you see there is some kind of correlation that when you have good TM score and good in the but actually look at that, you have great variety, huge variety. Okay, LDT, this is local, this is the implement is pretty good also with the TM score. In all and LDT works also. You see it increases with this one. So you see trends that make sense, but some do not make a trend at all. Okay, naturally, this is a class score. Class score has nothing to do with anything else, but it's an interesting point. Now I put with respect to the RMSD, you see the RMSD up to 60 actually, whatever it means. So you see this is in all decrease very fast. If the index is close to 0.1, you are down there. Afterwards, it decreases and disappears. Right? TM score is the same. So you have a very decrease in TM score, which that is expected. Now I will show you real data instead of statistics. So this is RMSD here. I stop at 60, all right, because 60 is already, maybe the next one is, this is there's no point looking at RNA, but you see that some are much higher, and that goes to 120. And here you have the TM, so you can put a Z bar 0.8 or 0.7. But you see many are also have very low TMs, and that is a different puzzles. And when they are low TMs, you see also that they are high RMSD. You see there are huge spread in RMSD. So some people are doing pretty well, and you see those at the bottom are very good. Huh? Some are doing very well, but some are really you have a huge spread. So we have many groups. We come back to that again. All right, now there is a question of template and no template. So, and that is very complicated with RNA because it's not only sequence. You can look at the sequence, try to map, see your blast or whatever, and say, okay, these two sequences look alike. Then you know exactly how close you are, and then how many mutations you are, and that is easy to copy something that exists. But it's not always this way. Sometimes it's more subtle than this because it is the 3D is the 3D is more invariant than the sequence. So it's not always easy to see from the sequence that you are in the same folder or not. But anyway, sometimes we know very clearly that there is a template. Sometimes we don't know but you see, I just show you without template, and without template is we know there is nothing existing there in 3D. For example, before for gold RNA, there was nothing. The first time it was solved, it was nothing. So gold is somewhere there. So again, your RMSDs are very without templates. They increase very fast. And some of them are okay, but here you have a huge variation in output. Now, what to do with these numbers? So here I put the RMSD, I put some kind of type of molecules, and you see also again the huge variation. This is the different puzzles, and this is the different groups, right? And the colors are just to separate them slightly. So you see sometimes that, for example, here, but there is a tRNA. That's why it is there, right? But so if you read PZ27 alone, you are up there immediately. That's the tRNA also. And then here, this is some RNA, for example, this is loopy type-like. Then you are okay. But for most of them, it's above 5, or even above 14 or 13. So it's really, from my experience, when you are above 12, 13, some indices are not correctly positioned, and you can have knots even. But when you are above 20, like that, most of the indices are not in the right place, and you saw more and more they have knots. And this the thread with the RNA inside the loop, and things like that is never seen to now. So we aim to find. We would like to have between 0 and 12. This is in this region. They should retreat, go down this way, just to have an idea. Again, distance between two phosphate is 7 angstrom. Clash-core. This is something that really irritates me a lot because this is not acceptable in 2025 because these atoms should not collide into each other. And with refinement program, the atoms we know the distances, we know the angles, we know the approach. They should not collide into each other. They should not. Normal X-ray structure or even cryo-EM structure, there the clash-core is way below 10 or sometimes very often below 5. So it's not because it's a computer output that you can have this type of stuff. Geometry is really key. What is really worse, I noticed recently, that RNA is chiral, is beta B ribose, but 1/3 of the submitted models present chirality violations. 64% of the groups that submitted models had some models with chirality violation, 43 among 3. Some groups had chirality violation for almost all of the submitted models. If you play around with the chirality, I mean, actually you will have a nice fold, you will go closer, but it's really false money. Everything you do you cannot trust anymore. And so most of the chirality violation occur at C5' when they link the phosphate, and actually you have the wrong one. Or at C1' when they link to the base, and so they put it alpha instead of beta, etc. This should not occur. All the tools are there to prevent this. Again, we know that if you increase the degrees of freedom by structural inversion, it's easier for fighting, maybe folding properly. You will get, but at the expense of native close contacts. That means if you have made a major error like this, all your native contacts are disrupted. You are losing them. So stereochemical violations indicate an incorrect local for a backbone pathway. That is the phosphate are colliding into each other on the So it's again the difference between accuracy and precision. So an accurate model with a low precision may have more potential, right? I mean, but for instance, function, but an inaccurate model with a high precision. You can produce very inaccurate model with very high precision. That is all the distances, angles, and no class score. That's it. But whatever approach you use at the end, what is delivered, the model should conform to accepted distances and geometry. So I will show you some example now. This is tiny RNA. It's a ribozyme, I think, if you see something like that. And now, so it's complicated. You have a lot of, so the green is always green is experimental, and blue is the model. And you see the RMSD is 5.4, if all is 68%, and TM is 26%. This is strange to me. This is annoying for me when I see these kind of things where the TM is very low and the inf is rather high, or inf is more sensitive. But you can see that, for example, the green pair, these two should be talking to each other, but you see they do not talk at all. Right? And here, the green should talk to each other. You see that the blue and they don't talk at all. So they are too far. They are in proximity, but they're really here. For example, this could be almost ready, but it's not there. Naturally, so at the end again, the fold is like this. The bases are somewhere, but they are packed in there, but in disorder. You know, it's like kids that you tell them to clean the room, and they put everything in the drawer and put the drawer. My room is clean. So it's about it looks like this. So it's a pity because they are so important. Because for example, A52, that is also the same ribozyme. This message group should go to that's the one that should work exchange. So the precision here must be very, very high to do catalysis. All right. And again, you see that even this base pair that is so critical, they even don't talk to each other. 52 is about right, it's a safe position, but the critical one, because they want to exchange with this one, they don't talk, they are too far away. It's impossible to understand the chemistry in this case. And here it shows also again a little bit what I was discussing with, if you superimpose that region here, the DOS nucleotide, consider the global structure, you say use spiral and you make RMSD or just fit it together, you will see this kind of far away. But now, if you take only these 4 nucleotides and you superimpose them, it's pretty good. It's much better. Also, one base pair is not there. So locally, sometimes they can get some kind of good, but it's disconnected from the rest of the backbone. This is an interesting case, very interesting, because more and more, recently it's very rare to find a new module, as I call them, a new building block that is seen in 3D. It's very rare. Here there is one. It's this one. I'll show you. You see the difference? You see GC base pair, right? But look at the backbone. How is the backbone? Can you see the backbone? Here, this is where is my little there. Here, 5 prime, 3 prime, right? Here, 5 prime, 3 prime. Then make what's a base pair? Flat and parallel. Two structures with this kind of things. So this is one of the rare occasions. And naturally, when you do the sequence, there was a structure. This one, it's SH5, which is an RNA element in some viruses. And so where you have it, you can align, but it's not easy to align. You align, if you know the 3D structure, you can align. And you can see what is there. You have this U going to this A, this C going to this G, that's nice. This GC pair is maintained, and the parallel stuff is here. Here is GG, GA, CGC, and going with this CCC, and there with this GCC. So they did manage pretty well for this. This is the new next one, but that was a difficult situation to predict with this new model. That's what you see here, because all these one one group managed pretty well with an info of close to 90%, but TM again 50%. I think some people, they try to improve the TM, they don't care for the rest. We'll come back to this. And this one that I showed you are just the very strong one that you see stressed in this you see in the deformation profile. Another example, very complicated. This is a dimer. You see that you have these two RNAs and they interact with each other. We see more and more dimers now, multimers, whatever. Naturally, this you have to have the 3D first, probably. You have to position them correctly, but then the loops that interact with each other should be also facing each other to make it. Here again, info is 0.7, term is 0.7. This is an agreement. This is in the model. I think so. And here, this is in the crystal where they talk to each other. All right, so you see that they are about the same place, they're in the same region, but they don't see the complementarity. I don't understand this. You would see something like this in your screen. You see, wait a minute, you have CCCG and there you have a GGC. Naturally, you have to pair. You have to look there, especially if they're in proximity. But naturally, maybe it's not so. Here, just to show you some example. So these are large group 2 introns or group 1 introns, the species 39. The blue arrows, TS04, this is alpha 43. And so it's some, for example, here. Higher is difficult work because there was no real homologous structures. Naturally, group 1 and group 2, you have plenty of homologous structures, and some people are doing pretty well. Some people, they work as if there is no homologous structures. Everything is beyond 45, 60s. Or here, you see that. But some people can manage. But alpha 43, but it's no better than anybody else. No better than anybody else, all right? And this is another one. They have two groups also. And one group is really more than 15, and another group is below. I managed somehow, but he is in the bad group. So there is no different really yet. Just I'm coming to an end. Then now just because Jerome is interested in. We did some kind of binding of small molecule. Here naturally we have a lot of aptamers, and here is an aptamer for guanine or something like that. And you have the crystal structure; it's very precise, etc. And here you have this is a crystal structure. This is the model 255. So you see, this is 269. The RMSD, they get it pretty right compared to that one. See, 268. So it's not 200. Sometimes they do make mistakes, but it's pretty. You recognize the binding. I don't know how to evaluate that. It's not very precise how to evaluate. Is this one better than this one? I don't know. Yes, he got this one. This one is, etc. But that here in this case, the RMSDs are 2.827. Here the RMSDs are 4.3, 4.7, etc. There is nobody is close. You see, there is very far away. They missed it. They all missed it. They didn't see it at all. Right? But this is also it's a minor roof, etc. So it's not very conspicuous. It's difficult to find. So I said, if you do not have, if your modeling already is not around 2.8, no point to try to talk a small molecule. Now, the way we compute the score is rather primitive, but just to show you a little bit, that depends on the number of targets, the number of how many. Here you have they submitted for 34 targets. You have there are 15 of them. They were in the 5 best, and actually the score end up is 49. So that's why we will call alpha 43 will be down there. That's a little bit one way of scoring, just to show you that you have 38 groups and you have 27 group below, so 28 plus 27 group, but 27 never in the top 5. So they are always far away from everything. Many groups are doing this. And again, here, that is the previous one, that you're human-based. We tried always to separate human-based and computer-based. The idea is that there is only a computer seeing the sequence and giving the output. And so the Polish groups with Marta Szasniuk and Janusz Koer, with RNA Composer and Zbigniew, they are doing pretty well. They submit a lot of them. Not everybody has the website ready to accept just a sequence and give back the. They have 2 days. Normally there are no humans see that. It's only we send an email to the computer and then they send back. Right? Here, naturally, they can do all what they want. So it's Jen, Pinchudas, Shashnuka, Bushnik, the same group of generally they are among the top, right? There, they can do whatever they want. It's human-based. So what I just told you, here we rerun AlphaFold 3 on all of them, and we are no pro. We just use it as in continual fashion. So we just and the red dot present the result, and you see it's very often it is in the middle. Where's my red dot? Yeah, it is there. Sometimes it's below, sometimes it's pretty woolly here. It's above again. Sometimes it's higher. So here you see it, for example, this is surprising. This is the TM. The TM are better than for the RMSD. So they must be adapting the model as a function of TM and not RMSD. But TM, that was the success also with protein. So up to now, are not different. AF3 prediction not different from the bulk of the other predictor. The results are worse for RMSD ranking than for TM rankings. I say they don't play the games in the way it's submitting to blind tests up to now. I mean, we need here when you submit there and you submit normally you see we know the structure does not exist. And here, when they do that, in most of their program, they mix training and test sets. We don't know what is in the training set, what is in the test set. And then there's some of them, they take our result from 10 years ago and they compare the result they obtain now with those results of the other colleagues 10 years ago, which I think is unfair. And then you cannot claim on performance in this way. There is lack of transparency because not all this is generally available. I think they should be more careful about this kind of transparency and telling exactly, which is not really easy to separate a good training set from a test set, especially with RNA. It's certainly not trivial. So I tried to show global fold is not sufficient, that you have many other parameters. Actually, coding versus million is not so easy. With cryo-EM, we have to be also more careful when we compare. So it's not so easy. We need maybe new tools for comparing, for assessing, because of they are approximate the way they do that is not the same than X-ray. Finally, we have no single comprehensive scoring function for assessing accuracy. It's a mixture of all of them. Inf is the only one where baseline is assessed and stacking is assessed at the same time. We can see no single prediction mission stands out yet. Sometimes some program from Riju is better. Sometimes it's from Janusz. You cannot tell which one will. I think in the future, I hope great hopes in AI, but they can generalize folding groups. But I think we all need to use systematic multiple second alignments at this. Look at them, it's much more detailed and more seriously. They do bring a lot of information, an amazing amount of information. All right. So I just want to thank Chicha in Guangzhou and his students and for the help and continuity over the years in this program, and you all for your attention. Thank you very much.
So we open the floor for question. Anyone?
So you mentioned that. Right now, like these like deep learning models are they seem to be kind of favoring different reward metrics for, let's say, like TM over like mean square deviation, the angstrom level. is this something that's kind of universal across all these different models that you're evaluating, like AlphaFold3 and even like some Rosetta-based algorithms? Is this like a consistent thing that you do see? Like they're just favoring one thing over the others and they're not really holistic?
I don't know. I noticed that with AlphaFold3, but I don't know about the others. Because again, I can only compare what I receive. If they don't send models to RNA-Puzzles, I can't. I'm not going to test their program myself on them. Yes. So because I want to be sure that the advantage of this type of approach is that the structure is not out when you receive it. It is not in PDB. It's not there, except sometimes if you have a sequence that is homologous to another one, but you say it's homologous. Homologous marriage is very precious. We need to be able to do that. But you see that a lot too many people are able to do it. You see, when I showed you this is an homologous structure, you could copy it exactly and change a little bit from there and there. But it's different type of programming than de novo. Right. So homologous modeling is very important, but you assess that differently from one first. And I don't know whether they do. I know that some people prefer TM because TM is never as precise as looking at the base pairs and things like that. But we need to be able at least to know they are close to each other. They could tell me, okay, they are close to each other, but I don't know what they do. But at least they are close to each other. Maybe explain.
Can I just follow up real quick? So you also mentioned that there's a high degree of variance between, I guess, like the accuracy and predictability amongst different submitted models.
Yeah.
Do you ever see this variance amongst like the ground truth amongst the actual like structure that people generate on the structural side? And if so, how do you reconcile differences?
Generally, they give us sometimes they give us more than one model. What we do when we have more than one model, it's not like in NMR when they give you a whole bunch. So it's just that's why we don't look so much with NMR. But sometimes they give 2 or 3 models. I say we take the best. So we compare with them all and we take the best one.
Like the structure.
But yes, we take the best one. We don't compare in between but generally it is small effect, small differences they see.
So it's not.
But I don't think you get this kind of variance that we saw there. There's RMSD of 60 angstrom or 30 angstrom. Yeah, and the diameter of an helix is 20.
Thank you.
Okay, that was a good point.
I think part of the question was also, have you had any entries where there were 2 structures, experimental structures?
2 experimental structures is sometimes, yes, with the same sequence, you mean?
Yeah.
Yeah, sometimes you have also monomers and dimers, yes. So we treat them separately.
And then
we tell them.
When it's an aptamer or there may be
with and without, we tell them. Yes.
You tell them they need that glutamine.
Yes, there's glutamine there.
Okay.
Sometimes the crystallographer or the structuralist doesn't want to tell, so we can give the empty one. Afterwards, but we have all these different types.
Because then it's a question without the
yes,
the glutamine, what is this?
It's not fair.
Yeah,
it's not entirely fair. Yes, very square.
Merci, Eric. I have a question. You found a couple of methods that are better than others in general. Have you thought about a consensus scoring, where you have multiple methods contributing to your model, and maybe finding, I don't know, a voting system, I don't know, but something that is more taking advantage of all of them? Especially, I found that some are good on random switches, some are good on aptamers, but they're not necessarily good on the same families.
I would love to, but this is it's not my specialty anymore. It's more computer people, bioinformaticians, and it's really tricky because these parameters that they do not have the same weights, the different same contribution. And there are people, so at CAS, they use a lot of Z-score. So but they put Z-score, and then they put 1/3 for that one, 1/2 for this one, 0.3 for that one, and you don't know where these parameters come from. Why did they put 1/3 for this and 1/2 for that one? So that's also fiddling. So but then you end up with 1 Z-score. Okay, but you can push one parameter or remove one parameter. So So for me, there is no solution yet. So this is an open problem. If somebody is interested, but it's really, I don't know, there must be ways to find out what would be the best score. But that's a big trouble.
If I can comment, I think this kind of approach, like ensemble approach, where we combine different approaches, would be interesting to be done, but maybe at the base pair level, not when you have the full sequence, because
I don't know.
it's too complicated.
That's why I'm asking.
Yeah.
But if you look at the base pair only at base pair level, you can take so you have inf. It is in the inf. We separate them. You have the inf for stacking. You have the inf for non-one-stacking. Even the non-one-stacking base pairs to predict them correctly, identify, annotate them correctly. When the models are full of class scores, of chirality problem, and what you get out of this is garbage, because the programs cannot handle a wrong RNA structure. So fundamentally wrong. So it's not easy to even to assess that. In most cases, they don't see any, so it's fine. But I cannot look at all of them by eye. I look at the structure wise, and then I know exactly how many base pairs there are there. But so the evaluation of this is not always easy. For the stacking, is easy. But afterward, you have to tell, okay, I'm just taking this. So the unique score would be a difficult goal.
I have a couple of questions online, but
Okay.
first I would like to ask them more questions from the room. No? Right, so the first question offline from Vincent was what about modeling before and after microRNA have interacted with the 3' end UTR? Are you able to model over long or short distances?
Well, this is a very difficult question because also this is for me the future in research here is RNPs. So to predict RNA complex with a protein is very, very difficult. I think it's even more difficult than with a small ligand, etc., because of all the diversity. And so microRNA bound to argonaute, etc. That is, I don't think the tools are there yet. Before we need more structures. The RNPs are very important, but it's very difficult.
There's another question that I'm wondering if an effort across RNA groups to train an AI, so for instance, a language model, with feedback from expert globally will train the model toward accuracy. Given that state-of-the-art AI model are using reinforcement learning techniques to improve accuracy, essentially feedback on the outcomes.
Maybe that's a good point. Maybe it's clear that I don't know a lot of it. What is important there, what I have been stressing or tried to stress, is geometry. RNA is geometry. Because the bases are planar already. You see in protein it's not the same. You are going all directions. Just this planarity, the fact that they can interact this way, they don't interact this way. Okay, they can interact, but they interact in the plane, or this way, but they're in the plane. So geometry is very important. Then it's bulky also. Helices are bulky. So the helices, they can approach, we know from which kind of part they are. It's again geometry. Can you train with geometrical mind? This is and I think a human, when training, we're helping to train, will always look with the geometry. This geometrical approach, coupled with sequence analysis. Can you couple sequences analysis and knowledge from sequences with 3D structure with geometry again? If you have I don't know, je sais pas. Voilà, la suivante, je crois que j'ai des aliments de séquence partout, mais je dois en avoir une. A sequence alignment, you know exactly. So when you see, can you tell this elixir, the green and the blue is really sharp? You see even the UAA there is invariant all the way. just the one before is not. Sometimes so there you have one nucleotide more. Can you train that? Can you train sequences? Can you together with a RNA 3D structure and link the two together? This, I think that's what you should do. Yeah, that's how humans could help to train this. We can do that. We can look at that and think in 3D.
Well, I think what it's complicated is if we do that, the reward. So when we train the model, we need to get the reward. So the reward would take so much time to get the reward if we go with human observation, that training a model on huge amount of data will take decades. It's where we currently, I think one of the blocker for that is good AI model works well when they have like a fast reward and they can train and win on the number of training amount. And I agree with you, having a knowledge informed training will be really better for the model, but it just looks like so much longer.
But don't you think you will raise? Suddenly the reward will be a big jump.
Yeah.
And so you start with small ones, etc., and you go maybe slowly. And it will be slow at the beginning, but very soon it will be a big because they will learn faster than us. But I think that's the major problem now. Yeah.
I think we're close from the end. But the good news is that for those of you that want to stay around, there is a networking session just outside. Eric and we staying around to discuss with you, and we're here for about 1 hour and a half, something like this. So I would like everyone to thank again for Eric for this wonderful talk.